everyone for joining us today. We have our own famous Dr. Sarder, who is known for his works in AI and renal imaging. Today, he's going to give us another talk on the same topic. Dr. Sarder, the stage is yours.
Thank you, everyone. Can everyone hear me? Yeah, well, okay, great. Thank you. So today I thought that, so far in the grand rounds, I talked about digital pathology, introduction of this topic. I thought that I would also introduce this emerging new area called Spatial Omics and how digital pathology, which is the pathology in particular, which is the gateway to making clinical decision or in clinical medicine, known to be, kind of, state of the art thing, right, for clinical work, right? How that makes this emerging topic called spatial omics for making foundational understanding of systems, as well as connect the information that these new technologies are providing from this new domain can be connected with clinical information. So before we start, a disclosure that I'm an advisor of this company called DigiPath Inc., which is a digital pathology company. So that being said, I would start with a bit fundamental question that our lab in biomedical microscopy or in biomedical medicine is trying to understand, which is the basics of many labs. Even in clinical domain, when you are looking at a slide, it is quite apparent that when we are looking at an image, right? Image tells us image is worth 1,000 words, as you can see. In a computer, that comes through various process like tissue section preparation on a glass slide. It goes through a microscope optics, preparation of the tissue, imaging. But the basic question and fundamental question that we ask, that how the information hidden in red, green, and blue pixels, right, that we measure here in the image, how this information, right, how this information gets perturbated or gets changed due to some sort of small minute changes in cells, as well as even at atomic level, for example, in DNA or RNA transcript level. And once these changes are there, what can we do to understand the minute information there, and how can we understand in a reverse way, like what is happening in this image? For example, in this case, this is a kidney, glomerulus image, where you can see this renal cells, podocytes. They are kind of perpendicular on top of the capillary. For a mouse which is moderately diabetic but has no sign of proteinuria or no symptoms, but it's still showing that change, right? So how we can infer that, right? So this is the fundamental, and in order to understand this fundamental, like, we take a very systematic approach. But before we start, right, I will give you some background for the people. Definitely this is nephrology department, and this is Department of Medicine. People would have background of what I'm going to talk about. But in case there are engineers or people who are present here from other domain, right, maybe from the philosophy department, let's understand few fundamental, right? So histology is nothing but a way of staining thin tissue samples, which are used for examining basics in tissue samples, or having better understanding of what is going on in a tissue sample. So histology is nothing but staining that tissue with chemical agents. For example, in this case, this tissue is stained by something called hemotoxylin and eosin. This tissue is stained by something called periodic acid strip, right? And it basically gives us some sort of contrast in the data or in the image, or maybe under the microscope, to have better understanding of what is going on, or underlying in the underlying disease, in the underlying tissue, to diagnose about disease, right? And what I wanted to know, Martel, I mean, if people are not familiar, these dark spots, right, these are hematoxylin stains. These are staining the nuclei, different cell nuclei, and there are other regions, right, which are like, pink or reddish, which are typically, staining the matrix, right? So that being said, what is digital pathology? Whatever I just said, that is very traditional way of, like when someone put a tissue sample after staining underneath the microscope, they look into that and they, like, pathologists, clinicians, they diagnosed by looking at the tissue, right? But one modern advancement in this aspect is this basically scanning this whole tissue samples using something called whole slide image scanner. This is not quite modern. It started from 2010, this trend, and we are kind of in an era today where we are still trying to get to the peak of this era of digital scanning. Although, in clinical context, these digital images, right, they are not still being used because of other regulations, regulatory need that are need to be implemented in clinic. But when you scan it, right, still for research purpose or wherever this is used as a fully digital way for diagnosis, it gives us an image like this where we can get the whole tissue sections when a small location maybe is this glomerulus that you guys are familiar with, right? So but I wanted to give you a little bit of comparison of how this data, right, it looks like when we compare with radiology and kind of data that we handle. So this is a digital whole slide image, right, and the amount of pixels, right, that is like 26,000 times 34,000 pixels, right? And one small locations of this thing is this glomerulus, which is 776 times 519. But in a typical radiology, in one section of the diagram image could be 512 by 512, right? So when in histology we are looking at this data, this is the amount of data we are talking about. And when we data scientists work with this data, we deal with this massive dimensionality hidden in this data to infer the massive amount of data from here, which may not be captured or may not be discerned well when someone is visually examining this data, right? And then I finally want to talk about little bit about omics. I know, like in clinical medicine, bulk omics based analysis, or when we go for some sort of diagnosis, like doctors sometimes send our blood samples for some paneling, right? So omics is nothing but a way to get similar information from tissues, from other samples, like liquid samples, right? But in a bulk way, means it basically measures maybe mRNA transcripts, it measures other information, protein information, but in a bulk way. And this is traditional bulk omics, where you take the whole tissue and you kind of get the information from the tissue without preserving the spatial identity. But we are finally in an era, this is a recent trend in last several years, last 3 4 years. This trend is called spatialomics. What spatialomics does? Spatialomics, these are like bunch of, or the ensemble of technologies, which I would say, which allow us to measure, right, omics information from a tissue samples via preserving the spatial information, or via keeping the spatial information intact. And what these technologies can be, one of these technologies called codex. It measures like 40 different markers in one time. From the whole slide image, you can see different things are popping up, and this is basically codex is basically we are measuring protein. This is a 10x vision sample where you see histology overlaid with this spots. Each of these spots measures thousands, about 20,000 transcript information. So spatially resolved transcript information. This is a deep data. MI phase, this measures targeted protein and transcript in the same assay. Then imaging mass spectrometry. This is very interesting. This has poor resolution, but it measures what? It measures the metabolomics informations, right? In UF we have a leader on this area, Dr. Ramon San. He is spearheading this area. So then imaging mass cytometry, right? Then it's another way of measuring different cell types. Then multiplex ion beam imaging, Cosmex. It's basically targeted hundreds of transcript data, but in a special way. This is another new area with maybe with higher precision than other previous methods. And then 10x Xenium, which is my favorite, which I have been working on these days, and my we are funded to generate 10x Xenium data from kidney tissue samples from 100 tissues. And what 10x Xenium does, it measures transcript information at single cell level. As Heisenberg's uncertainty principle says, that moment we reduce the resolution, we try to get more spatial information, we have to reduce the amount of transcript that we can measure. And there is a new technology that just came up. It's called Visium HD. They are saying like they can get like about 10,000 transcript information from single cell level. I do not know the details of chemistry behind them, but I am amazed that where the technology is. And this is the right time where we want to understand where how the digital pathology which is used in clinic and how they are connected with some of the rich information these special technologies are providing us. Then that being said, for engineering student, kidney background, this department needs no brainer. We humans has two kidneys. Each kidney has which are useful for filtration, and it's a very interesting and important organ for us. And each kidney has multiple this kind of units which are called nephrons, which starts with something called blood vessel that comes here in form entangled loop for filtration. It comes out and then go back to the body after filtration. And this thing is called glomerulus. Then you have tubule, which basically can carry the waste like that gets filtrated here, and then take a major role in absorption and reabsorption. It goes to the collecting duct through which the urine gets excreted out of the body, and there are several such tubules. And to give a complete picture, when we talk about pathology, right, this is one cut, 2D cut of this histology tissue, which shows, right, I mean, different areas of these kidneys, right, these nephrons, right, this is glomerulus, that's how you see, right, this is the tubule, right, what this is one of the tubule in 2D cut, what you see about this 3D system. Then you have see arteries, right, remember that vessel is coming inside, and then the blood is flowing through this, and getting this this filtration, and then the process of reabsorption and reabsorption for this tubule. So this is artery, and then there are interstitium, right? Interstitium is nothing but the open regions, right, between these 3D structures, right? So that being said, finally, I'm ready to talk about the overview of what I wanted to, talk about our research. And just to recapitulate, as I mentioned in the beginning, the interest here is what do we understand fundamentally about the tissue, cell and tissue interaction in a system just based on RGB pixel data. And in order to do that, we take a very systematic approach and learn this overall system using AI machine learning you in from in varying level of scale from 10 micron at cell level to 200 micron, which is at the functional tissue unit level. So I will talk about how to get understand about single cell, estimate that from bright field histology, how to estimate multiple cell types, how to segment functional tissue unit, and relationship between cell and functional tissue unit. And this is the fundamental to understand biology for any organ system. And we are just using at this moment kidney as a vehicle, and we'll show at the end of this talk how we are trying to reach out to other organ systems to make it more generic. To focus more, we'll start with solving this, very interesting problem. Imagine you have a histology image which costs maybe $10 to $20 to generate in a clinic, right? A histology image from a tissue sample. We'll use deep learning model, machine learning model to do to estimate measurement, which would otherwise call multiple thousands dollars from a histology image to generate, that is understanding how we can precisely estimate multiple cell type, and or what it would take to precisely estimate multiple cell type. Why? Again, we wanted to understand cell tissue interaction and model that as explained in that first slide, which is the fundamental central hypothesis, central goal of this lab. We are heavily motivated. We are kind of, heavily motivated, trained, taught. We are the student of this program called Human Biomolecular Atlas program, which has a goal to create an open global atlas of the human body at cellular level to do this fundamental understanding, not just in kidney, but in everywhere. And it is spanned with 42 sites in 14 US states and 4 countries, and it's divided with 5 categories based on the type of work being done. This consortium, definitely, the one effort is the computational effort that we do to do data integration from at various scale, from various technologies, from different part of the body, and this is called HubMap, infrastructure visualization and engagement effort, which is called Hive, and this is supported by tissue mapping centers who generate tissues based on multiple various technologies, and other efforts. And I'm very proud, and I'm very fortunate and blessed that I'm the PI of one of the Hive component, and we spearhead the work of doing this connection with bright field histology and bring that expertise from bright field histology domain to this hub map. And I have a multidisciplinary team of researchers who are coming from various domains, from analytics, usability, data generation, even training of next generation, as well as from software engineering. And we together try to have this basic understanding of the system as a collective effort as part of this community called HubMap. And I think let's try to understand and string our problem to understanding annotation of single cell type. Single cell type, I have a histology image and I want to predict one single cell type. What is the challenge? In order to understand and appreciate the challenge, this is a small tissue image, like in a glomerulus image. It has this nuclei, cell nuclei, as discussed, that those different cell nuclei. And one challenge is that if I ask each of you to count this number of cells and then mark which cell type is what, right? It's a fun game, right? And all of us, I think this is an nephrology department, right? I mean, it would be fun, and this is an expert who has done that for me. By now, by the way, I can do that also for kidney, at least. I mean, I'm kind of a fake pathologist these days, so I can do that for kidney. But the whole point here is that in order to have that understanding, what I said in the beginning, in that first slide, I wanted to do that in a big data context to understand some of the hidden structure which is not even discernible by human eye, right? And think about that. In order to do that, I need this kind of ground truth data. And if I have to generate this ground truth to train my model, to it ultimately teach the computer to how these cell types are, this is a very difficult work, right? Because ground truth generation itself is very difficult, because you can imagine that it is a problem of precision. Second problem. Second problem is these tissue samples, right? These are 2D cut of a 3D tissue. So whatever I see as a podocyte on top of a capillary may not be on top of the capillary when I see that in an image. It could appear somewhere in the middle, right? And this is an example. This is a PA stent tissue sample, and this is immunofluorescence level ground truth, which shows where the podocytes are. And I asked renal pathologists who are expert renal pathologists to level where the podocytes are, and they level the podocytes, the peripheral, very well, but they miss the central ones, right? So this is the fundamental problem we are talking about. And one seminal contribution we did, right? If you go back to 2021 November issue of JSN, our work appeared on the cover because we, for the first time in the literature, has shown the feasibility or pipeline of how to predict a single cell type from a bright field histology image, right? So So what we did, we took paired whole slide, histology and immunofluorescence image, and then from the immunofluorescence image, we labeled the podocytes, right? In this case, we took podocyte. It's not like, we can ideally use any cell types, but, we had the podocyte, we work on the podocyte, and we work on the podocyte for a region. But then we trained the computer and we predicted that from the whole slide image, these podocytes, right? Like, where they are located, and what is important here is the same section histology, which is the key, and we can discuss a lot about why we need same section histology for training this kind of model, which we kind of mastered for several years. It's kind of apparent, but in the literature for in AI tasks, this was not there. And we obtained reasonable sensitivity specificity, right? And then we went back to this data, right, where the manual annotation showed only the peripheral podocyte, and where original IF showed clearly manual annotation was missing some detail. Then we looked at the output of our model, and which was able to predict those central podocytes, so which was a success. That being said, I wanted to open your mind, because this is a grand round. That question is, are we really counting, right? And you can imagine that counting becomes really complicated when we look at tissue samples which are thick, right? Because it's always a 2D projection, right? When you go from thin to thick, number of cells, they change, right? So we have to be careful about that. And even when we train an AI model, it's just not just training an AI model. You have to understand how the lab medicine or lab workflow works. The other thing, this is an unpublished data from one of my champion colleague, Lingyan Shi from UCSD, and Dr. Sanjay Jain from WashU. And this is talking about 3D histology. Maybe time to talk about 3D data, 3D histology, to improve our cell annotation from histology. This is being done from this, it's SRH leveling, it's Raman leveling, it's Raman, microscopy is being done here to reconstruct this 3D image, but we are moving to that direction. That being said, while we keep our mind, thinking about, how we can go to that direction, let's talk about how we can scale this work and try to annotate multiple cell types, right? Which sometimes I thought is a magic, but then I thought this is an Operation Moon, Operation Mars project that we wanted to solve. So basically, I have a histology image of kidney, and I want to tell how many cell types are there and what type of cells are there, right? And I have been working for this project for last 3 years, and we are still working, right? And I will show you some systematic approach in how we do this and where we are going. So we take a whole tissue samples, right? And then we use this special technological codex, right? And then once we obtain the codex image from the same section, we get the histology H&E image. Then we run the codex image using a tool called VTEA, which clustered the cells and mapped the cells back to generate the ground truth. Once we have a ground truth, we have a same histology image and ground truth cell types, and we train the model to find out where the cells are. And this is where we are today. That we have 14 sections. In 3-4 years, I was able to only generate 14 sections, which are really, really good with respect to their corresponding level. But we have mapped 2.5 million of cells while doing the work, and which are precisely mapped ground truth, and which took us real effort in last 4 years. And we are testing cell types is 500,000. We are there in a balance accuracy of 0.73. When I told my student, look, we need to reach to 0.8 to publish in Nature, or in a not Nature, in a high impact journal. Baseline is 0.1 because we're trying to predict 10 different cell types here. But my student yesterday asked me, hey, are we reaching to a plateau, right? But how we do this? This is not like just, we take a machine learning model and we just apply it. It required quite a bit of systematic engineering approach. So we first, in order to do that, we try to take all the different cell types, right, in a very granular way, and we throw that into the system and see how we are doing and where is the confusion. We ask the question, if the confusion is somewhere, right, which cannot be explained biologically, or if the confusion is somewhere where the biological it can be explained. For example, like cell types, right? confusion between collecting duct like cells, like primary cells and intercalated cells, right, which look very similar, right? So even if you like try to separate them, they can never be separated. So what we do, once we try to understand that, what we do is merge cell types, and we can say, okay, they cannot be separated, right? And we merge and we come up to this 10 cell types where we obtain 0.73. But we are still finding, confusion between immune cell and endothelial cell, which are not correct, because, they are biologically, morphologically very different, but we are getting the confusion. And we are trying to, getting at the bottom of it, how we can, add more biologically informed feature to train our model to get that job done. And we hope that this accuracy would be zero point 0.8 in the next few months. The other problem is the ground truth. We talked about that. Sometimes we predict, but we don't know the ground truth, right? For example, this is a prediction, but we don't know the ground truth. And that thing also reduces our accuracy. We applied these techniques in intestine. In intestine, right, our accuracy is balance accuracy is 0.47 against a baseline of 0.08. We are trying to do the same thing, predict the cell types from histology. We are trying in skin, and we are also trying in lung. And as we speak, like in a very soon, in a month from now, we'll hope to be able to at least predict 5 different cell types from lung histology, right? And where we are going, of course, that would be a separate story, a whole new project with multiple organ types, and I can come up with that. But I think for now, I would talk about the ground truth generation. For the ground truth generation, for this kind of project, right, we need cell segmentation. Cell segmentation means DAPI segmentation, which is at the bottom of this image of this kind of task, which is very fundamental. There we have seen so many algorithms. People talk about cell segmentation algorithm. We have our own algorithm, and this part has to be correct to segment DAPI from a whole slide image in a systematic, in a generalized way. We extract features. With moment the DAPI is segmented, they are all registered. We extract feature from all the channel. We map them back in 2D to find out what type of cells are there, and depending on marker abundance, right, we call for cells, right? So this cell type mapping takes our 80% of our time, if the prediction takes 20% of our time. So this is so important. That being said, I have a very junior student, so who took the challenge of predicting this information for the case of spatial data, where histology has high resolution, but spatial data is not so high resolution. But at the same time, spatial data provides a deep data. So that is 10x Visium technology. As you know that when it uses spots, right, they are like little far apart, and they provide like 20,000 transcript information, right? And those 20,000 transcript information can precisely tell us like the not exactly cell type at the spatially at single cell level, but at population level. And this student, right, went little far, and he tried to show based on this work, like how good engineering principle, how intuition can be applied. So what he did, he took something called foundation model. Foundation models are like little buzz these days in digital pathology domain, right? So what foundation models does, they take millions or millions of data from like 40,000 whole slide images from digital pathology, or they take corresponding text, clinical information, and they train the model such a way that model learn everything about the data. So what my student did in a very intuitive way, he took all these functional tissue unit and he poured that in this algorithm with text prompt, right? Whether it's coming from glomerulus, tubule, interstitial vessels, and it created some sort of a dictionary, which basically tells us whether this thing is really a glomerulus or not, but using text level information. So once the glomerulus is there, right, he generated a text prompt, right? A brief text prompt that says, okay, whether this is glomerulus, and if this is glomerulus, and this glomerulus has the following cell types are present, right? Because why I want to do this way, because this opens up a possibility possibility to do a better engineering. Imagine if I have a cloud tool available and I ask you guys to write text and have your understanding, right? This kind of system can be trained based on human interaction and find out rare cell types based on human observation, while I can also bring spatial information. So that's where he is going with his intuition. And I was so amazed by like looking at the result. And he's actually going for his PhD qualifying exam today. And he wanted to be in this meeting. I told him, no, 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 go back home and come dressed and do your exam. That is more important. But he came up with this idea. Although his accuracy is, although it's higher than baseline, it's not still there at 0.8 level, but he found something biologically significant when we compared his tool to compare reference versus diabetes, where we can immediately cross validate, and we found that in diabetes the podocyte content is going down based on his prediction in the test data, and ECGC, endothelial cell content, is going up. So that's how we found that he is kind of in the right direction, but this work needs tuning. And I wanted to specify that because this was a very intuitive, very interesting work that he did, right? Its name is Jamie Farmin, by the way. It's a new PhD student. I wanted to learn a little bit about what I presented in last year's grand round. This is just in one slide. This was my first 5-6 years of faculty job, where I was segmenting structure in kidney tissue and trying to understand. And right now this structural segmentation is like a technology in my lab, right, where we can segment like all these different structures, right, like glomerulus, tubules, interstitial fibrosis. It's basically any structure, peritubular capillary, TBM. And remember that we also work as in collaboration with other groups. So peritubular capillary is coming from my great colleague Laura Barisoni. They have developed that and they have provided us the model. But that being said, why I wanted to talk about this FTO segmentation, because remember the first slide, what I said. I wanted to understand from histology the interaction between cell and tissue to have a fundamental understanding about human biology using AI in a big data context. That's the central theme of my lab and my career. And we wanted to expand in other tissue types, and we'll show some results where we are going and bringing more and more biology to make some fundamental contribution using these tools. So that's why this tissue segmentation is important. And from the tissue segmentation, imagine moment we segment it, we can ask the computer to find out several information: how big the structure is, how many nuclei are there, what type of other information is there. And you can imagine each dot here is a structure, right? And they have multidimensional this feature which we can project, and that may allow us to find trend. For example, you see like trend from one angle, you don't see a trend in the data, but from this angle it shows three, right? So this is called like the manifolds, understanding of the manifold, right? So how that understanding of the manifold is compared with the cell and a cell leveling, that's what is something that we wanted to do. But that being said, since I talked about this FTU segmentation, right, functional tissue unit or FTU segmentation and feature extraction, I wanted to show some of the work we have done that shows the importance of this features. And one case study is diabetic nephropathy, where we know that current state of the art is Starvard classification, where doctors look this tissue underneath the mouth. They mostly look at glomerulus and changes in glomerulus to level the tissue in different classes, right? So this is a seminal contribution we did. This is my first PhD student, Brandon Genley. He is in Johnson & Johnson right now. We published this in JSN in 2019. We have close to 200 citation in this paper by now. And he apparently recently came back to me and he said like, he want to also be involved in research in my lab as a volunteer, because he's working things in Johnson & Johnson, but he's not getting happy with with the research part, or maybe in a sense like he wanted to do more research, right? So what this is what he did, which is very intuitive. It's a whole slide image, right? And the way doctors level it, right, what they do, they go to each of these structures and they'll actually level what is the level or the grade of this tissue in diabetic nephropathy. So go to those structures, they say some changes, they say okay, this is grade maybe 3. Then here they come and they say, okay, this is grade 4. Maybe when they come here, they remain to the same decision because the change here is as same as this. So they don't change it. When they come here, they keep it as it is, as the old one, because this one is not as damaged. This is looks like a 2A change, but 3 is higher, right? So that's how they level it. Since we have a computational tool, now computer knows how to segment all this. Computer knows how to extract the features automatically. Then we use something called recurrent glomerular classification. If you remember how Google tells you these days how to end your sentence, right? When you are writing something, it's the same thing. Data comes in series, and it continuously learn a level. And we have shown using this work, we can mimic the same level of accuracy the doctors can achieve in this work, right? Using our features that we develop, right? Or that we designed. Second case study is transplant outcome prediction. So we took like about 29 transplant surveillance biopsy cases, and of course I have a better result than this. I think one of my student is working on that. She's using like thousands, hundreds of transplant biopsies, but in this case we have transplant surveillance biopsies. These are from 1 year transplant, and from each this biopsy we segmented the glomeruli, right, this functional tissue unit, and from this glomeruli we extracted these features that we talk about. And this is a UMAP projections of this data. Each dot is a glomerulus in a low resolution, and then when we clustered it, we found one cluster, right? When we overlay that with EGFR decline, poor outcome versus bad outcome, one cluster came out to be highly predictive of the bad progression. And for that cluster we found features, right? Those features came out to be important for for progression, right? And that's how we showed the importance of our data. Then going back to another example, that another case study is comparing amyloidosis versus diabetic nephropathy. Right. So we know these two are maximal change diseases, means like, both of are like, nodular changes. Both are affected by nodular changes, but both of them have maximal changes. And question is, if we talk about nodular changes, how can we find out something that would, based on our feature, so that we find out like, they are still distinct, right? Because it may not be apparent for someone who is not coming from pathology. So what we did, we compared these two data sets, right, from this disease with minimal change disease, to create a classifier using features which would apparently tell us there are nodular changes. But then we use all the features to refine that classifier and ask the AI to tell us that once we define that classifier, what are the features that are abundant, right, in each of these classes. And we found apparently diabetic nephropathy that has more cellularity than amyloidosis, which is basically AI is telling us based on features that, okay, we can get something which that human eyes may not be apparently tell us, right? And consequently we figured out the data from those corresponding regions, right? And we found the changes, right? We saw like how they look different. So AI can pull that information out. So that being said, we are part of also another prestigious consortium called Kidney Precision Medicine Project, and I contribute with our tools for image data segmentation and feature extraction. And it's a no-brainer. Kidney Precision Medicine Project, it's a multi-year collaboration for ethically obtaining kidney biopsies from participants with AKI, CKD, and reference. And to understand better AKI and CKD, because we know that diabetes is still a fatal disease where we only go by management. There is no like a pill that comes and that tells me, okay, it just reverts back, right? So the Kidney Precision Medicine Project is trying to collect such biopsies from those patients who are not clinically indicated for much earlier, and generating this multimodal data, means histology and spatial data, and trying to understand this fundamental understanding to understand new biomarker that can be painted for understanding about the disease. And we are spearheading to develop this pipeline for kidney. Our group actually is spearheading this pipeline, like, to segmentation of the tissue samples, visualizations, and making a sense of these different, structural features which we are working on. And we are also contributing to Kidney Precision Medicine Project Atlas, where if you go and download the data, you will also see for the general public not only the data, whole slide image, spatial data, but you can also download the segmentation mask and pathomic feature which our lab is generating and contributing to the community for the bigger goal of the community. Without, we haven't submitted our paper yet, but the community is able to download this data and study this data with respect to other feature. So that's where we are contributing in KPMP, right? Our bigger goal is to, stratify patients, right, based on these features using other set of data, for example, clinical data, spatial data, and find out who are in risk. And then finally compare this information with the spatial biology data to see what's going on, to show further like some of the, some of the most interesting thing that can be done, or some of the fascinating thing that can be done based on our derived features. We took about, hundreds of biopsy samples generated by KPMP. These are all CKD, chronic kidney disease biopsy, where with each of these dot is tubules, and we took their features what we measured based on our work. And when we looked at that features in low dimension and applied a technique called tubular trajectory prediction, we found there is a precise trajectory of how this tubule changes, which has some relationship how the tubular trajectory changes in biology to understand acute changes to chronic acute changes to chronicity. And we also found certain information in that trajectory which are not defined. Our goal next step is to work with spatial data, right, to understand the meaning of this trajectory, which we are working on right now. So finally, I'm at the final stage of the presentation. I think we have like about 7 minutes so that I can have time for audience to ask questions. Is now, remember in the beginning I mentioned that we want to have this understanding of from atoms to anatomy, cell level understanding across scales, which is a very, very engineering way of approaching, to a problem, but to do fundamental understanding. And this paper, fingers crossed, we submitted to Nature Methods yesterday, and if Samuel Border is present, he single-handedly work on this project. I don't know whether he is attending or not, but he can talk about this paper for hours and hours. I'm not sure whether I don't see him here, but that's okay. So what he developed a tool, online tool, right, where we put all our tools together and we systematically visualize and analyze using AI the relationship between cells and functional tissue unit in a dynamic interaction between this, and to have an understanding of their relationship. So this is one, this thing of the tool where you can see the histology and its overlay with, different functional tissue units, and it shows some sort of a map of the cells, right? And link to the demo, you can see a demo of the tool. It's available via this link, and the tool can be accessed via this link, right? You can go there. We are recruiting users. We have more than 100 users of these tools. This is one example. You have a histology sample. The tool would show you the cellular abundance, right, of according and spatially, right, with respect to histology. And we have, not only it does that, it also does quantitative analysis, right? It shows the cell composition for different structures, different regions. It does morphological clustering of tissue samples. Compare that with the spatial data, you would be able to download this data. There are like a large set of samples we made available with this system, which you can even analyze and download using our AI system and write, fundamental papers on biology, right? And this is one of the fascinating thing what we observed. Like this is a CKD kidney biopsy, right? And we found like these are the chronic changes, right? Using this tool, what the chronic like the glomes are changing, right? But you see like this immune cell infiltration, right? And the load of those immune cells, they are also there in this glomerulus. But these glomerulus that is like already changed, right? And that doesn't have this information. So our tool is finally is there to do that fascinating biology, right? Of like, we have some sort of junction in a tissue where we have, some sort of, inflammation happening, and then immune cells, they are going and to try to see what's going on. And then those the acute changes, they are finally transferring to chronicity, right? So that's what we found that this tool is capable of doing based on AI. And this is where we'll focus in coming years, and hope to work with some of you who are working in fundamental biology by bringing our tool. We showed that we can separate AKI, CKD, and reference tissue samples using our tool. Each of these dots, they are like different patients. They are with their aggregated features in UMAP space, and some are from AKI, some are CKD, some are reference. But the tool is also showing the cell abundance, right? How AKI and CKD, they are different than reference, right? With respect to fibroblasts, with respect to endothelial cell content and podocyte content. We are using new technologies. Our tool is capable of showing information for more technologies which operates at single cell level. Our tool also allows to do cell type annotations using codex data, right? And we have a usability study going on, which is recruiting people from various domain, starting from residents, students, to pathologists, to nephrologists. Sign up for the usability study. We have this link, and so far we have more than 100 participants, and we are trying to work on a paper based on participant. And this is like a real participant who is using our tool, right? So this is like and they are writing a question answer to how they feel about using this tool. At the backbone of this work, we developed this thing called comprised. This is the backbone of any of our front-end development. So whatever you saw to have this tool called Fusion to do basic understanding of cell and tissue, this is the front end, but it uses at the back end something called comprised or computational renal pathology suite. What it does, the goal of this system is to take histology slides and other data, predominantly for histology slide, and in a public facing way. And it uses the storage as well as CPU and GPU node in a HPCC framework, and use all those horses, and in a very, very secured way, right? Because academic institutes, when they work with us with this kind of system, or immediately they get scared, because we are in a health system. So security processes matter, how secure this system can be. So we work on this for last 2-3 years to make this system as secure as possible. And now this system is running in Hypergator. We have a public facing one available. You can go there and test it. But we have a more functional, with a high amount of CPU and GPU capability, a tool available which uses more computational resource and heavy computational power, which can only be accessible by using your UFID and password. And someone else from other institute, they can use their in commons setup using their university login and password to access this system, which we developed for image segmentation. But it can run all those AI tool, and it is at the backbone of whatever I just presented. That being said, I mean, it can be established in any HPCC setup, and it has a link to the demo, that link. But I think I don't have time to go to the details, so I will skip all these slides about what is the scalability, what are the standardization of this thing, to make it as to follow the best engineering practices to develop this tool, right? I would just wrap up. I think we just showed a way of engineering pathomic features and show that the value and the explainability of those features. We showed a way to prediction of cell type and state from histology, which is challenging, but we showed the path and the approach, how you systematically do that, how you can use biologist knowledge to build these systems. We talked about fusion, we talked about end-to-end systems. And these are not the fundamental research I do. We are funded in three different areas. I hope that next year I will have enough data to talk about one of these to get to more fitting ground for this audience. We have the RO1 in diabetic nephropathy, kidney transplant, and lupus nephritis. And we all know that diabetic nephropathy, it's a disease where we still don't know how the early level changes, they are correlated with some of the clinical information. Kidney transplant, we know how many discards we have in kidney transplant domain. How can we use AI to do a optimal patient, recipient, and donor matching? And if my student Akshita is here, she is spearheading this work. Lupus nephritis, we know that how fatal those drugs are, right? Those drugs, they're painful, right? But how can we use these technologies to that I discussed? Using fundamental understanding to have a drug response done based on images. I've been closely working with Ben Sickle to generate a fundamental model to solve this kind of problem, where we use histology image as well as pathologist guidance, where someone like you, right, or an user, they can kind of interact with this system. And my goal is to define a disease by saying, for example, in diabetes, and someone mentioned like in diabetes, 2 out of 15 patients are always cannot be explained. I wanted to say how diabetic this person is, how much disease you are with a vector. So it's a continuum. That's what we are trying to model, right? That being said, other projects, I don't have time, but I wanted to quickly overview. We work on cardiac CT segmentation with a hope that someday, and the same thing in radiology, I'm trying to expand, right, with a goal to merge, because human disease is just not at cell, cannot be discerned at cell and molecular level, but can be discerned at a bigger level. One set of measurement could be enough. I wanted to expand this lab with radiology data analysis, which we are doing right now. We are working with Dr. Brian Ho's lab in cerebral aneurysm project to understand aneurysm and treatment response. Duet is a microscopy technique which uses H&E slide to measure fibrosis. And moment we talk about fibrosis, it has implication in kidney, maybe also in cancer. So we are expanding in that domain too. Fatima is, if she is here, she is spearheading, and she brought this concept of domain adaptation to to make sure, like when data comes from different lab, her tool still works in prediction of fibrosis using duet data. And it's not like we don't work in cancer. I have contribution in neuroendocrine tumor, and I would love to look at some if kidney cancer data available. Finally, we are in a stage where our lab has enough manpower to look at cancer, where in neuroendocrine tumor, we work on this famous problem called Ki-67 indexing to develop an automated method. And I'm part of also this consortium, which I never talk about, but I'm also part of ArtNet consortium, which is a cancer consortium where we are developing, the resistant the data for for patients or mouse model which are resistant to therapy, and we have like multimodal data we are collecting, which would soon be used for some of our project. So that being said, we are also, as part of our UF mission, we are heavily engaged in education and training undergrad or underprivileged, underrepresented students. And in collaboration with Yulia Strekalova, we hosted several events, spring research experience as well as summer research experience. We are submitting educational papers to understand students' perceptions, right, to how they take this kind of multidisciplinary educations. And we found a very positive results with a high statistical significance, where they found that these multidisciplinary approach, cross-disciplinary, cross-scale, is something like the students are approaching as the next generation to be Renaissance scientists or revolutionary scientists to bring new medicine concept. That being said, we have a high vision. Sky is the limit, or maybe sky is not the limit. It's there. We have the foundation that we built, and we are at the right moment standing to. Rome was not built in one day, but we hope that our tools have that we develop set the pillars across the scale, and we hope to work on some national level. Program centers, grants as soon as possible. Maybe O'Brien Kidney Center. Only thing is that in order to have O'Brien Kidney Center, I need to have all gray hair, and maybe I need long beard with all gray or something, because you need very very senior faculty, and we'll see what we can do. But even people in geology department, they come to us, and we have a funding with geology department. They get some sort of rocks, right, which is some sort of compositions which we basically find use our tool to understand what's there. Thanks to my collaborators, I have very good amount collaborations going on our lab, thanks to my students, trainees, and there are spaces open always. You can see, right, someone can just chip in. It doesn't matter what stage of career you are, our lab is always open to do science, and we always talk about science. That is what only I want to talk about, and I like to talk about, right? So we have spaces. We are fortunate with funding, but you never know, right? When these things get depleted, we need more and more money, more power, more energies, right? So please come join us, and I think we can make a difference. Thank you very much.